So thank you, Katie, for the introduction. Like she said, my name is Penny, and I'm a PhD student in the Keel Reef Lab, and I'm working on a molecular-based method for microbial source tracking within Mobile Bay. So coastal waters can experience elevated levels of fecal contamination, and this can be a result of human populations, unsupported infrastructure, and terrestrial watershed drainage. And because these coastal areas receive and drain a lot of water, pathogens can be conveyed to these waterways from a variety of sources. So you can have human sources like septic tank failures and sewage spills, and you can have non-human sources like wildlife, domesticated pets, or livestock. And this poses human health risks through contaminated fisheries, so like your filter feeding organisms like your oysters, as well as your recreational waters. So it's not uncommon to see beach closures because of high levels of fecal indicators. And this is because you don't want to ingest those pathogens. So whether it be because you're eating the contaminated fishery or you have like a wound and you're going in the water and it's going through your membrane. So areas that have these high levels of fecal indicators are really hard to manage. And this is mostly because when you have high levels, you don't know if it's just one contributor or if you have a lot of different organisms that are contributing to that. So this necessitates advanced approaches to manage and protect community health. Microbial source tracking, or MST techniques, try to identify microbial contamination, but there's a bunch of different methods and they all vary in capability and effort. So traditional microbial source tracking methods, or what is most commonly and widely used, are not able to distinguish source. So they can kind of only tell you that there are fecal indicators in your sample area, but you don't know where it's coming from. So this makes management difficult. But thankfully, molecular methods can tell you the biological source. And this is very important in our area because species-specific contributions to microbial indicators in Alabama waters are largely unknown. So the aim of my project is to identify major sources and drivers of microbial contamination within Mobile Bay using these molecular source tracking techniques. And I have two molecular techniques that I will be using. So the first is quantitative PCR, or qPCR. And this will be able to identify and quantify fecal-associated microbes. And it can do this by targeting genetically distinct strains of Bacteroides. Bacteroides is a gut bacteria, and it has such high host association that you can tell from its sequence whether the Bacteroides came from like a human gut or from a cow gut. So I can look at a sequence and tell you that that bacteria came from a cow without having to interact with the cow, which is very important. And for this, I have 6 different assays or markers. So I have a general marker, all Bacteroides. This is not as specific as my organism-specific ones. So this will pick up all of the different ones. It will pick up human Bacteroides as well as the cow Bacteroides. So this will serve as my total marker. And then I have 5 species-specific or organism-specific markers. I'm targeting human. I have a pig marker, which also picks up feral hog, which is really important because they're invasive to our area. And I have a dog marker, a cow marker, and a fowl marker, which picks up both chicken and duck. And as a caveat, qPCR markers are highly location specific. So these change depending on your microbiome and where you are. So these are I am testing them for work in Mobile Bay, which is a very important thing. To make sure that they're not cross-reactive and that these primer sets work in our area. And then my second molecular method is environmental DNA, or DNA metagenomics, and this will actually be able to identify all bacterial and DNA-based biomarkers within my sample, and it will provide some ecological context. And I know everyone knows about eDNA, it's great and amazing, but I want to do a little side note to clarify what exactly I am doing with my eDNA. So eDNA is DNA that's present in the environment. It could be blood, tissue, fecal matter, or bacteria. And to collect an eDNA sample, you just collect an environmental sample, so typically sediment or water, and you analyze what's in that sample. And there are 2 ways to analyze that. So in the top panel, it's showing a specific method for eDNA where you're using a primer set and you only want to see one species. So if you're looking at fish, you only want to see walleye, you can only see walleye in your samples if you use those walleye primers. Or you could do next generation sequencing, NGS techniques, and look at the entire community. So they've got like a little, I don't know, maybe a tuna, Lydia would know, a shark. You're looking at everything that's in there. You're not picking out one specific organism. So I am doing next generation sequencing. I'm doing metagenomics. So I'm looking at everything that's in my sample and I'm not pre-selecting for anything. So this is important because I can use this without knowing anything about my system. With the qPCR markers, I had to have some prior knowledge. I know that there's cows in the area. I know that there's pigs in the area. But with eDNA, I don't have to know anything about my study site. I can just go and collect a sample, which is very awesome. And eDNA metagenomics has not been implemented as a microbial source tracking technique. So, highlighting. I told you about my two methods. Now I'm going to tell you about my samples. So we have 13 sites that are all around Mobile Bay, and they represent potential fecal inputs to the Alabama coast. So at all 13 of these sites, we collected water samples on a 4-week basis over the course of a year. And at the 3 sites that are orange, we also collected oyster samples and sediment samples on a seasonal basis. So to clarify this, I'm going to walk you through this figure to clarify my sample matrix going from left to right. So we have our sample site that has got our water. It's got oysters that we put in a cage and tie to a piling or a dock, and sediment. And at all of these sites, all 13 sites, I collect water in a 10-liter sterilized carboy on a 4-week cycle. And then at those 3 sites that were orange, I collect oysters. That is a blender because we did have to blend them. And I collect a whirl pack of sediment. All of these samples that I collect get both of my molecular methods. So all 3 sample types, all time points, everything gets eDNA and qPCR. The only thing that's a little different are the oysters. So instead of straight eDNA, because if you do eDNA on an oyster, you get oyster, which is pretty intuitive and a little bit of a waste of money. So I'm focusing on the microbiome and using 16S rRNA to pull the microbes out of the oyster. And in addition to the Demetriades qPCR on the oysters, I will also be looking at E. coli. So to get into my chapters a little bit, my aims, my first one I think of as my qPCR chapter. So I want to quantify influences and sources of fecal-associated contamination in Mobile Bay using those Demetriades host associations. So specifically, I want to look at species-specific contributions and see if there are any spatial or temporal patterns to what I'm seeing. And I also want to look at environmental drivers. So when we went out and collected our samples, we also collected water chemistry, weather data, discharge, whole host of things to give us context to what we're seeing. So I want to see if any of those environmental attributes are driving the concentrations that we see. My second chapter is more eDNA dominated. So this is the bigger picture of investigating fluxes and drivers of the microbial community composition and function in Mobile Bay. So I want to look at the sediment and see if it's a source or a sink of these fecal pathogens. I'm going to assemble MAGs, which are metagenome assembled genomes. And by doing this, I can look at the genomes of the eDNA sequences that I collected, and I can annotate them for pathogenic pathways and identify viruses. And using these eDNA sequences from a microbial source tracking perspective, I want to fill in data gaps from the qPCR analyses. So because I was so specific with the qPCR and I picked out my 5 species that I want to look at, the eDNA will hopefully pick up other things that I can use to fill in those gaps. And I also want to look at taxa change with oxygen concentration and nutrient concentration, the dominant nitrogen forms. My third chapter is my oyster chapter. So I will be utilizing only molecular-based techniques to look at the oyster microbiome. Specifically, I'm going to look at how the concentrations of Demetriades change between sediment, water, and the oyster samples. And I also want to ask the question, can Demetriades source tracking be done in oysters? And what I mean by this is, are the relative contributions of my species the same between sediment, water, and oysters? Or is there some kind of preference that the oysters have that wouldn't make them a good sentinel for source tracking. And then I'm also just going to delve a little into the microbiome of the eastern oyster. I might do a method comparison with that between Illumina and Oxford Nanopore. So that might be cool. And then aim 4 is my toolkit chapter. So I mentioned previously that eDNA has not been used as a microbial source tracking method. So in order to have other people easily implement it into the field, I'm creating a toolkit and I'm hosting a workshop for community partners and stakeholders. The workshop will be April 25th, 2025. You are all invited. And this toolkit will take stakeholders from sample collection through filtration, DNA extraction, sequencing, and bioinformatic analysis to help them implement eDNA as a microbial source tracking tool. And I will also be surveying the participants before and after the workshop to get an idea of their knowledge and retention and the usefulness of the workshop and the toolkit. And I'm hoping that this will be a nice unit that others can use in different areas. So just to bring it back to my broader impact, this study will be the first to identify and quantify fecal sources on the Alabama coast. And it will pioneer the integration of next generation microbial source tracking monitoring efforts, which is the eDNA. And this will allow system scale management. Molecular-based methods can lead to improvements in the management of fecal contamination and stability and growth of the local oyster industry. And data will support shellfish aquaculture and seafood safety, safe swimming, tourism, and ecosystem health. And since I mentioned data, I have some preliminary data to show you all. It is qPCR of my water samples. And I'm going to walk you through the structure of this slide because the next 4 slides will be set up the same way. So on the left-hand side, we have my little cartoon of what my qPCR marker is. The bottom is just like a general sentence, a general takeaway for you to get from the slide. And then there's going to be a bar chart of all of my qPCR data. So the left axis is quantity of the marker. For this, it is the all Bacteroides marker, so my general or my total marker. And some of the graphs have breaks in the y-axis because I wanted to visualize some of that like intersite variation because the quantity is so different in magnitude. And not all of the graphs have the same y-axis. Not all of them have the same quantity. So keep that in mind as I'm going through. The horizontal axis is site, and then it is color-coded by month that the sample is taken. So this is the all Bacteroides marker. And what we can see from it is that it's pretty prevalent at all sites over all time points. And I want to reiterate that Bacteroides is non-pathogenic. This is, do not be concerned, please. But it's prevalent at all sites over all time points. We see a little bit of an increase in the summer months, those like gray, bluish guys right there. And then this site right here, West Mobile Bay, is high all the time. That was collected at the effluent of the Mobile wastewater treatment plant. So makes sense that it's high. It will also be a really nice guide for me as I'm going through my eDNA sequences because I can use it as a marker of wastewater. All right, now we've got our humans. Human was also found at all sites and was the major contributor to allback. So it really follows that trend and accounts for the majority of what we see with the allback. Not very surprising. Then we have our PEG marker. The PEG was also found at all of the sites, but it's a bit more episodic. So we see a lot less colors on the graph. We see a lot of May. Don't know what's going on in May, but there's a lot of pigs in May. And then we have our foul marker. It was only found at a few sites, but it's pretty consistent at those few sites that it was found. And our dog marker was also found at most sites, but it's really, you see seasonal dependence in this. Like it's very much pink and purple, and that's fall. So I haven't yet dug into any of the stats. That is next, but I'm very excited to see why or what this means. And then our cows, I did not make a chart because it was only positive at 2 of the points. So it was positive at site 10 in December and site 4 in March. So yay cows for not pooping everywhere. And then I also ran regression tests to compare my total marker to my species specific marker. What I found was that human and fowl were significantly correlated with the total. So these are the regression plots that I made with the R squared. And again, the human accounts for the majority So as I mentioned, preliminary data, just got it. I'm working on it. And going forward, I will be doing a bunch of multivariate statistics on my qPCR data to analyze those trends and identify those drivers that I was talking about. And the environmental attributes, some of the ones that I really want to focus on are the freshwater discharge data, the water chemistry, and the weather and physical conditions. And my eDNA sequences, I have been bioinformaticsing on the computer. So I've been working on it. It will take a little longer than the stats, but lots to come. So I would like to thank my PI, Dr. Brandy Peel-Reese, my committee members, Drs. Jessica Jones, Ruth Carmichael, and Sinead Hine. I would like to thank the members of the Curies and Carmichael Labs, my funding sources, our EPA grant stakeholders, and thank you guys for listening to me. Yeah. So that's a good question, and that's why I'm looking at all the different sample types. There are a few studies that have come out recently looking at the decay rates of these bacteria and their markers in the sediment. So like, since I'm doing eDNA, if the DNA is there, is it actually viable or is it active? Or has the bacteria just kind of like fizzled out? And there are some studies that have been trying to move away from E. coli and Enterococcus because they have been found to persist and become a part of the natural flora within the sediment. So hopefully these more specific markers aren't part of the normal flora because we can tell the source that they came from. I will answer the second part of that first. Oh, sorry, I will repeat for online. Dr. Powers wanted to know if 3 sites is enough for the oysters and what I think they will tell me differently than the sediment and the water. So we picked 3 sites because we wanted to collect sediment and oysters and water at the same site instead of having them all differently. And not all of the sites have a sedimented bottom, and not all of the sites can support oysters. So those ended up being the only sites that worked. And I do think that the oysters might show something differently because they're filter feeding and it's not just like transient. I think that they might, I'm honestly really interested to see if I can source track within the oysters, like if there will be a difference in the species specific contributions to the oyster. Like, I don't know if they're going to select for pig or something. Like, that would be really cool to see. So I'm interested in whether they're doing something to the microbial indicators. Yeah. So we homogenize the whole oyster. Sorry, guys online. The question was, am I analyzing some specific microbiome of the oyster, like gut microbiome, or am I doing the whole thing, which I am doing the whole thing? We homogenize the whole oyster and the liquid. Thanks, Mackenzie. Cool. Yay.
Thank you, Penny. Okay, so next up we have Lydia Hayes-Guastella. Lydia did her bachelor's in microbiology at Michigan State University, where she was researching microorganisms surviving in extremely high pH fluids in terrestrial serpentinizing systems. She then went on to do her master's in marine biology at Texas A&M University Corpus Christi, where she focused on nitrogen cycling in restored and natural wetlands influenced by wastewater treatment plants. So now she is merging her interests of microbial life in extreme environments and biogeochemical cycling through the pursuit of a PhD in marine sciences under the advisement of Dr. Brandy Kilreese. So come on up, Lydia.
Doesn't know how to get.
All right. Hi, everybody. I'm Lydia Hayes-Guastella, and I am a third-year PhD student in Brandy Kilreese's lab. And I'm going to talk to you about my project looking at microbial carbon cycling in energy-limited subsurface environments. So prokaryotes and fungi represent two of the largest carbon biomass budgets on the planet, with 77 and 12 gigatons of carbon, respectively, which makes them essential for global biogeochemical cycling. And while they are very important to global biogeochemical cycling, there's not much that is known about them in the deep subsurface marine environments. And when we talk about deep subsurface, we're talking below the entire water column into the seafloor. And if you can see all these little dots on this map, that represents all of the sites that have been drilled for scientific ocean drilling by International Ocean Discovery Program, IODP, and all of its previous iterations. And what's important about this is that in nearly 60 years of scientific ocean drilling, only 4% of samples have been studied for microbial communities. And even fewer of those have looked at microbial fungal communities. So there's a lot to learn about the microbial deep subsurface. And why is this important? You might think that the deep subsurface is too extreme of environment. There's not going to be a lot of life down there. But in the entire ocean water column, there's approximately 1.2 times 10 to the 29th cells. And that's more cells than there are stars in the entire universe, which is estimated at approximately 1 times 10 to the 24th. And with that being said, there are approximately the same number of cells in the entire marine subsurface as there are in the ocean water column. So it makes them pretty important to study. So with that, my overarching objectives are to elucidate the roles of fungal prokaryotic microbial communities in biogeochemical cycling, and to ascertain the different metabolisms and adaptations that can aid in survivability of these communities. And I'm going to do that by looking at two distinct energy-limited marine subsurface environments. The first one is the South Atlantic transect, which is a transect of a spreading segment from the Mid-Atlantic Ridge. And the second site is the Mariana Forearc serpentinite mud volcanoes, which is part of the Mariana subduction zone. And I'm going to address these objectives using two different methods. The first is culture independent methods, which means that I don't have to grow things in the lab in order to look at that data. And the second is culture dependent methods, which does rely on successful cultivation of an organism. So going into my methods further, my culture independent methods, I'm going to be using metagenomics, which is all of the DNA in a sample. So similar to what Penny was talking about with her eDNA. And what metagenomics can tell you is it can tell you the community structure, so all of the taxa that are in that sample, and it can also tell you the potential function of the community. So what types of pathways exist in the genomic signature, but not necessarily whether those pathways are active or not. And so we start off with our sample, which in my case is either a sediment core or a filter that has had filters or fluid filtered through it. We extract the DNA and sequence that. And after some bioinformatics, we can clean and assemble and bin those sequences. And from that, we can look at taxonomic classification. We can look at gene quantification and the potential function of those organisms, and you do some statistical analyses with our environmental parameters. to look at significant changes based on environmental contributors. And then metatranscriptomics, which is all of the RNA in a sample. So the difference from metagenomics to metatranscriptomics is RNA will tell you what is actually active in the sample. So what types of metabolisms are actually being utilized in the sample at the time of the sample that the sample was preserved. And the method for this is fairly similar to DNA, where you start with your sample and you extract the RNA. You'll then reverse transcribe that RNA into complementary DNA. And then through sequencing and bioinformatics, you can look at the transcriptome annotation, which tells you exactly what genes are in the metatranscriptome. Look at taxonomic quantification, functional quantification, and then also mapping metabolic pathways of active communities. And there are precautions that we use when we're working with these samples, which include working under a clean room and using aseptic technique, which includes wearing full PPE, including shoe covers, lab coat, gloves, face masks, and hairnet to really ensure that when we're working, my samples are very low biomass, so we want to make sure that the cells that we're looking at are from the environment and not from us. And so going on to my culture dependent methods, we start off, I'm using this to look at fungal communities. We start off with either a sediment culture or sediment sample or a borehole fluid and inoculate that into media, in this case, 25% potato dextrose, and we monitor those for growth. And once we see growth, we transfer that into a larger media bottle with antibiotics so that we're not bringing along any bacterial tag-alongs. And once we have enough growth, we'll extract the DNA and sequence that for their internal transcribed spacer region, which is a taxonomic gene marker for identifying fungal community members. And once we have those sequences back, we compare them to internet databases, which allow us to find their closest known relatives. And from there, we can do literature search and find what types of pathways their closest known relatives utilize to degrade carbon and other types of metabolisms. So we can look for novelty of these organisms, but also what their potential is to go forward with whole genome sequencing and ecophysiological testing. So whole genome sequencing will allow us to see what a specific organism has the potential of doing in the environment. And then our ecophysiological testing will be able to tell us its growth tolerances at different pHs, salinity, oxygen levels, temperature, their ability to degrade recalcitrant carbon, and also growth at different carbonate and bicarbonate ratios. And you might think, how do we confirm that our isolate is from the environment that we're studying and not from contamination? And we do that by looking at culture independent amplicon sequencing of the original sample. So we will sequence the ITS community of our original sample that hasn't been cultivated in the lab to confirm that our cultures exist within that community. And all the precautions for this are very similar to the previous slide, with the addition of all culturing is done under a UV sterilized laminar flow hood over an open flame. And so going on to the merit of this project is combining culture independent and dependent methods to look at both prokaryotic and eukaryotic microbial communities in the marine deep subsurface. It's going to focus on the structure and function of those communities and their role in carbon cycling at an ocean spreading zone and a subduction zone. It'll be the first study to cultivate fungi from either of these sites, the first to examine the microbial community structure and function at the South Atlantic transect, and the first to look at the microbial community function in borehole fluids in the Mariana Forearm. And so I'm going to move on to the specifics of the first site. So the South Atlantic transect was drilled by International Ocean Discovery Program during expeditions 390 and 393 in 2022 on the drilling vessel Joides Resolution. They drilled 5 different sites for microbial study. The sites closest to the Mid-Atlantic Ridge on the eastern side, the deepest sediments are approximately 60 meters below the seafloor. So that's, again, talking below the entire water column and then 60 meters into the seafloor. And then the sites closest to the South American continent, the deepest sediments were approximately 300 meters below the seafloor. This area represents an age transect where the sediments closest to the Mid-Atlantic Ridge are approximately 6.6 million years old, and furthest from the Mid-Atlantic Ridge are approximately 61.2 million years old. This also represents a carbon transect where the areas closest to the Mid-Atlantic Ridge are highest in sediment carbonates and lowest in organic carbon. And furthest from the Mid-Atlantic Ridge are lowest in carbonates and highest in organic carbon. And that trend continues with depth, where the sediments closest to the surface will have highest organic matter and furthest from the surface have highest carbonate concentrations. And we expect that the microbial community structure is really going to be driven by this carbon structure, where areas that have highest carbonates and highest inorganic carbon and lowest organic carbon are going to be primarily fungal. And then as you have higher organic carbon, there's going to be more heterotrophic bacteria. All of these sites were drilled on the same slow to intermediate spreading segment of the Mid-Atlantic Ridge at approximately 31 degrees south. So they're all in the same spreading segment, so it's all from the same source of the Mid-Atlantic Ridge. My objectives of these 2 chapters, first is going to be to analyze the microbial community structure and function, again, using culture-independent methods, which include the metagenomics, which again is all of the DNA in a sample, and then metatranscriptomics, which is all of the RNA in a sample. And then the second chapter will be to synthesize the roles of the fungal members of the microbial community on biogeochemical cycling. And that's going to be through the culture-dependent methods, looking at fungal isolation, whole genome sequencing, and ecophysiological testing. And going into my preliminary data for these sections, I have successfully extracted DNA from 2 of the 5 sites, and I'm still working on the other sites and RNA extractions. But we have successfully isolated 25 fungal cultures. And after preliminary ITS sequencing, I have chosen 2 organisms based on their closest match on online databases and their ability to degrade recalcitrant carbons, which are carbons that are difficult for other organisms to break down. And the organisms that I chose are isolate number 14, which is closest match, is Fusarium oxysporum at a 96%. And its closest match has been found in literature to have the ability to degrade lignins, aromatic compounds, and cellulose degradation. And the second isolate from this site is isolate number 24, which its closest match is Cladosporium species at a 93%. And its closest match's ability to degrade recalcitrant carbons include cellulose degradation, chitin degradation, lignin degradation, and polycyclic aromatic hydrocarbon degradation. And going on to my second site, Mariana Forearc serpentinite mud volcanoes. So this system is part of the Isobonin Marianas Arc Trench system. It is a non-accretionary convergent plate margin that is undergoing active serpentinization, which is a water-rock reaction that produces high pH fluids elevated in hydrogen and methane. It has high alkalinity and is very sulfide rich. And when we're talking high pH fluids, these fluids are 11 to 12.5, so extremely high pH. We drilled 4 serpentinite mud volcanoes, and each of them have a cork or cork light installed, and that stands for a circulation obviation retrofit kit. And what that is basically a giant metal straw that has been stuck inside of a drilled borehole down into the seafloor. And what it allows us to do is access deeply sourced fluids without being contaminated by seawater. And so we sampled in 2022 on the RV Killamoana using the ROV Jason out of Woods Hole. And we were able to sample borehole fluids as well as surface sediments through push cores. And we have an additional expedition planned in December of this year to go back and sample more fluids. So the objectives for these two chapters of my dissertation are to determine the metabolic diversity of fluid and surface sediment microbial communities, and that's through culture independent methods. And for this, we'll be focusing on the metatranscriptomes, which again is all of the RNA and the active functional genes in the system. And then the second chapter main objective is to synthesize the role of fungal members of the microbial community on biogeochemical cycling. And again, that's using culture-dependent methods similar to the other site with fungal isolation, whole genome sequencing, and ecophysiological testing. And for my preliminary data, I don't expect you to read this, but I just want to note that we have been able to successfully extract quantifiable RNA from borehole fluids at 3 of our 4 sites. And we've successfully extracted RNA from bottom water, which is bottom seafloor water, at 2 of our 4 sites. And then our return expedition in December is going to allow us to go back and sample those same sites that we weren't able to get enough biomass the first time around and make sure that we are able to get it. And for my fungal chapter, we successfully isolated 7 total organisms from these high pH fluids, and we are moving along with 3 of them based on their potential carbon degradation ability. And the first one is a most closely related match, was a Stomiopeltis species at an 88%, so it's not very related to anything that has been found in this genera. But in literature, its closest match has been found as a saprobe, meaning it's able to degrade recalcitrant carbons on plants in China and Thailand. But no organisms in this genera have ever been sequenced for a whole genome. So we don't really know what they're actively able to do for recalcitrant carbon degradation. The second organism is our third isolate, and it is most closely related to Cladosporium species at 97%, which in literature has been found to degrade cellulose, chitin, lignin, and polycyclic aromatic hydrocarbons. And then our third isolate at this site is most closely related to a Pneumoniopeltis, which is its closest match was a 95%. And this organism in literature has been found to be able to degrade xylans, but also to use polycyclic aromatic hydrocarbons as a sole source of carbon in culture. And with that, I would like to thank Dr. Brandy Keel-Reese and the Keel-Reese Lab, as well as the science parties and captain and crew on both expeditions that have allowed me to work with the samples I'm working with, and all of my committee members and my funding sources. And at this point, I'll take questions.
How actively what you like figure out what
Yeah, so the question for the people on Zoom was that I didn't mention much about the culture dependent organisms, and have I actually successfully isolated anything? And yes, all of my fungi that I talked about are all isolates that we have actively growing in the lab. Yes, so far we have cultivated them and we have extracted DNA and sent them for whole genome sequencing, but the next steps are to do ecophysiological testing, and I'm planning to look at pH differences, temperature, salinity, light and dark, oxygen dependency, and as well as different carbon sources and different sources of bicarbonate and carbonate ratios in their media. And okay, so the first question was, these organisms are growing in sediments on a geological timescale. So how do I expect, or what is the growth rate of these organisms, and how long do I expect it to take me to be able to do my growth experiments? And I haven't done growth curves on all of the organisms yet, but I do have some initial ones from my South Atlantic transect sites, and it takes, under normal atmospheric conditions, it takes approximately 3 days for growth to be visible, and then for growth to taper off, it was about 2 weeks. So they are growing at a pace that is reasonable to be able to do these experiments. And for the second question, the question was that the organisms are found in really high pressure conditions, and how do I expect, or that can't be replicated in the lab easily, and how do I expect that to affect my experiments? And with fungi, they are usually in spore form in sediments, so they're a little hardier. So the difference in pressure coming up, they're not likely to lyse as easy as some of the other cells, like bacterial cells. And once they're in culture, they're pretty stable. So their ability to grow at atmospheric pressure is, if you can grow them, then they'll grow. And since we already have some isolates that are growing, then that's what we're working with. Yes. Oh, okay. How do I look at that? So the question in the chat is anyone on your collaborative team characterizing the types of organic carbon at our sites? And yes, there are people for both sites that are looking at organic carbon. Yes. Mhm. Yes. So the question was, since we're going back in December, and we weren't able to successfully get RNA from all of the sites for the first round, and we're going back, are we going to be taking an additional replicate at all of the sites, or just the ones we weren't successful in sampling? And the answer to that is we are taking samples again from all of the sites. And we had, on our first round of sampling here, we had some problematic issues that we were not expecting, and now we can anticipate those, and we've planned accordingly to be able to combat those on our second expedition. So we plan to resample all of the sites and sequence as well.